putting together this global collaboration, somebody mentioned that I should talk to him, and it was incredible because he automatically connected me to everybody in Italy with all the emails and and all the introductions, and suddenly we had quite a significant input onto that study just because of that one meeting with him, and he really made it happen. It was incredible, and hearing today from Sylvia more about his past, I think I understand now why he was so loved and respected around the world. So starting with the top 10 papers, I mean, I was asked to talk about what's relevant for clinical management, but I kind of have changed the title to top papers in HIV, just because I'm also a clinician. I'm not only a clinician, but a scientist, and I think we mustn't focus only on clinically relevant things, because things which are dreams now are going to become the reality in the future. So we must remember what's happening in the lab as well. So, but just to start on the clinically relevant things, 2 drug regimens versus 3 drugs. I mean, this is obviously, there have been some game-changing papers. The much-awaited GEMINI study, which most of you probably already know, so I will discuss this relatively briefly. This was two separate trials with 48-week outcome data. That was comparing tenofovir FTC dolutegravir versus 3TC and dolutegravir. 700 patients in each group, and this is the kind of just going to test this pointer out here. Oh yeah, okay. So you can see here that the over 48 weeks, you can see very little difference in the virological response to both regimens, which is truly remarkable. I think you'll agree. This was carried out in 191 centres in 21 different countries. So a very, very significant study. Here you can see some of the data, so greater than 90% response rates in both groups. Virological non-response was very low at around 3% in both arms. And if we now move on to some of the treatment failures, you can see that at 48 weeks there wasn't any emergent integrase resistance, and indeed there wasn't any emergent NRTI resistance. Of course, this is only 48 weeks. We need to see what happens further down the line. And of course, in real life, people, especially in different settings, will be failing for longer periods without monitoring potentially. So we have to remember this is clinical trial data, but it's really, really good news. Dolutegravir in second line, I think this is really important, and there was, of course, the landmark study. One of my interests, of course, is in resource-limited settings, and I think we should all be aware that we do need better second-line regimens. At the moment, it's mainly boosted lopinavir in most places, and I think that tolerability is often a problem. So, Dawning published this year showed that this is a randomized trial comparing 2 NRTIs with dolutegravir versus 2 NRTIs plus boosted lopinavir. And this figure shows you that overall DTG in fact outperformed lopinavir, which wasn't very surprising. But what I think is interesting here is you can see if you break this down by viral load, you can see that viral loads below 100,000 copies, this is where you see the real difference between them. But in fact, if you go to viral loads above 100,000, they're pretty similar. So I think this result wasn't particularly emphasized, but I think this is important because it shows that potentially boosted PIs actually are possibly a little bit better at high viral loads, and you really only see the differences in tolerability when you're at low viral loads. So this is very good news because DTG is being scaled up. It's very cheap to cheap to manufacture in sub-Saharan Africa. So and will be used as first line, and is already being used as first line in many places. And these are much needed data for use as second line regimens for those who failed non-nukes potentially. If you just look at the table down here, you can see that here we go. Most patients entering had been treated with around 60% have been treated with tenofovir-based regimens, but significant proportions were on thymidine analogs when they switched to DTG. That's important to know. And in terms of resistance, at baseline, 30% had K65R, and importantly, there was very high rates of M184V or I, so a lot of lamivudine resistance and significant tenofovir resistance at baseline in this study. What I thought was interesting about the study as well is that we observed in 2 patients the emergence of G118R in integrase, conferring significant resistance to this drug. One was a clade B and one was a clade C. Mark Weinberg and others had reported 2 patients previously who had been switched to dolutegravir monotherapy in Canada and who had also developed G118R. And they put this down to polymorphic residues, meaning that you only needed a single nucleotide change to confer, to generate that G2R mutation. So I think that although the prevalence is relatively low, I think this is something that we need to be looking out for in the future. And of course, it leads to significant fold changes to dolutegravir that then in vitro were shown to accumulate more mutations and more resistance. So that's 2 drug regimens and second line use of dolutegravir. I thought it was important, given we're in the era of next generation sequencing, to present data on how useful this is, because the big question is, what is the clinical utility of NGS? And Roger, sitting here, along with other colleagues, has done a lovely study published in Lancet HIV this year, along with the PASA consortium, including Raf Hammers. And what they did is took the PASA network, which is a network of sites in sub-Saharan Africa, Nigeria, South Africa, Kenya, Uganda, Zambia, and they had something like 400 patients included, and looking at baseline drug resistance by an NGS. And what they were looking for is resistance at different thresholds, 20%, 10%, 5%, and 1%. And here you can see, and what they looked at was the impact of these mutations at different thresholds on outcome, response to first-line NNRTI-containing regimens. What you can see from this, hopefully, is that and you have to remember that Sanger detects pretty much at 10 or 20%. So imagine 12% being Sanger. By going down to 5%, you really don't make a huge increase in the sensitivity of detecting clinically relevant drug resistance that has an impact on outcome. And if you look at this thing called the diagnostic odds ratio, you can see that it's not particularly different at low thresholds, suggesting that NGS is adding a little bit of extra information, but not a huge amount. And this is really important to know, because I think a lot of us have been asking ourselves what the added value of this technology is. This graph down at the bottom here just shows they performed a case control analysis to look at this, the added value of NGS. And you can see here in the gray bars, the blue bars show what you would find with Sanger, and the gray bars are kind of essentially showing you what the added benefit of NGS is. So you can see it, in some cases it's significant, but in most cases it's relatively minor. So that's the utility of NGS. Next thing I thought was quite important and interesting is what we do with MDR HIV patients. There is a pipeline of new drugs, but it is still relatively sparse, you may say, and I think host-directed therapies are interesting, so not targeting the virus with drugs, but potentially targeting the host. We already had this model with CCR5 antagonism with Reservoir and other drugs. So I was quite interested by this paper on ibalizumab, which is now, I think, approaching being licensed in the US. So this was a phase 3 study using this monoclonal antibody against CD4, and importantly, this is a monoclonal that binds CD4 but does not, it's non-competitive, so the point being that CD4 can still signal via its usual interactions with the immune system. So that's the sort of thinking behind it, and this was given by, you see here, this drug is given as a loading dose intravenously, and then every 14 days, so it's every 2-week infusion. And in this study, they took people with triple-class resistance, 30 patients in the study, and half of patients had at least a 1 log drop compared to baseline, which is good, I think. So it's at least achieving some drop in these heavily experienced and highly drug-resistant patients. And interestingly, in those without response, they were able to find changes in envelope V5 that conferred in vitro reduced susceptibility to iberalizumab. So this offers some hope to individuals who have very few options left. So on the antibody note, there's been a lot of interest in the use of monoclonals for treatment of HIV. And this paper in the last couple of years, there have been papers of monoclonals being used in the humanized mouse model from the Nussensweig lab, and this year they published a paper of using combination BNAbs or broadly neutralizing antibodies in humans. So this is a paper in Nature last year. And what they've essentially done is taken 12 patients who are suppressed on antiretroviral therapy. They gave intravenous infusions of 2 BNAbs, 3 BNC117 and 1074. And this figure essentially shows you, so on the right, if I start with you on the right, these are the individuals recruited to the study. You can see that they are nearly all male, and they have been on antiretroviral therapy for quite some time. So the shortest was 2 years and the longest was 11. So these are suppressed individuals. And when you look at the data, they broke the results down by those who had what they called a resistant reservoir. So when you clone the envelopes out of those individuals and test them against the antibodies in vitro, there is no neutralization because of obviously sequence variability with an envelope. You can see that if you take patients who have got what they call resistant viruses, after analytical treatment interruption, everybody rebounds. But if you have a sensitive virus here, you can see that in this Kaplan-Meier analysis that most people rebound, but some don't. So this shows a sort of partial success. So overall, 2 patients out of 12 maintained suppression beyond 30 weeks after stopping antiretrovirals. So this is encouraging, but it's not. I mean, it's a proof of concept study in humans that combination monoclonals can actually suppress HIV, but of course it's only a small fraction of those who are treated. So it's a good start, but there's some way to go with this, I think. The next paper is a follow-on from the previous one. I thought this was quite important because, of course, those patients have their antiretroviral therapy interrupted during the study, and as I showed you, most of them rebounded. So, there's a big interest in where virus rebounds from and what's the characteristic of a rebounding virus after treatment interruption. And this study was a nice opportunity to study those re-emergent viruses. So, this paper in PNAS that followed was aiming to describe what the origin of rebounding virus was in those patients treated with the dual monoclonal antibody therapy. And so the take-home message really is interesting. So they studied these patients. So there are 12 patients here, and they performed near full-length sequencing, which is in brown here, and then they did a viral outgrowth assay where they take CD4 T cells from those patients before treatment interruption, and then activate those cells and look at what kind of viruses are coming out, and then sequence those. And then they looked at the plasma rebound virus here, in pink, and they've done a distance map, as it were, so looking at the genetic distance between different viruses. And in each patient, you can see that the important thing here is that there's a big overlap in the viruses that come out from the VOA assay, which is the outgrowth assay, and the full-length sequencing from PBMC. So those two, I mean, and in many ways this is unsurprising because you're sequencing from those peripheral blood T cells, and you're also outgrowing virus from those T cells, so they should be pretty similar. And in fact, I guess what's more interesting is that the overlap is only very small. So there are many viruses that don't look anything like the ones that grow out of T cells. So the sequencing and the outgrowth assay are not fully concordant. So I guess that's interesting. But I think what they found, and I found most interesting, was that the rebound viruses in the plasma completely different. I mean, there's absolutely no overlap genetically between what's coming out in the blood compared to what's in the cells. So this tells us, it's another piece of evidence that when we test PBMC in the blood, looking at the reservoir, it really doesn't have much correlation with what's going on where replication is happening, which is in the gut or lymph nodes or wherever. So I think that was quite interesting. But actually they then, and this phylogenetic tree just shows something pretty similar, where here you've got the rebound sequences in red. This is plasma virus in one patient. This is what happened at rebound. And all of these are the clones they found in cells. So this is cellular. All these guys down here are cells, cellular associated sequences, either by QVOA or by near full-length sequencing, and this is what comes out in the blood. So there's obviously a distance there, and you can see this is the distance marker here. So that's just another way of illustrating those data. Then they do some fancy phylogenetic analysis to look for recombination. I was not able to evaluate that assay, that method personally, but recombination analysis is notoriously difficult, and maybe let Anami discuss this later, but in any case, they performed a recombination analysis and found, or reported in the paper, that the reason that there's this big separation is that what you're seeing is recombinants coming out in plasma. So the re-emergent virus is not completely different, it's just a recombination. There's a recombination going on between sequences in cells that lead to this emergence. So that leads to the interesting possibility that there are multiple rounds of replication going on before you see the virus in the blood, which I guess makes sense. When you stop treatment, things don't just pop out from blood cells, that are floating around. Actually, what happens is that the virus is released in tissues, has to replicate a few times, and during that time recombines during those rounds of replication, and then the virus that emerges in the blood is a, let's say, fit variant or a highly replication competent variant, but is a recombinant of what's in cells. So that I think is really it's generated a really important hypothesis, I think, in the scientific field for us to further explore. So these are not conclusive in my view, but these are very important for us to direct us to the future. So prevention, we must talk about prevention in any discussion of important science, I think, especially given that there are high rates of new infections globally, and especially in certain hotspots around the world. So this is a paper from Hanneke Schutzmaker and Dan Baruch, testing a preventive vaccine that was consisting of a mosaic HIV vaccine. And this was a phase 1/2A trial that was both in humans and in rhesus monkeys. So this, just to tell you a bit more about the vaccine or the arms. So this is an adenovirus that has HIV proteins inserted into it, and the proteins encoded are GAG, POL, and envelope. So 3 of the major proteins, and some patients were then given an adjuvant as well as GP140 protein. So this is a protein. So you're actually putting protein into these vaccines as well as a virus encoding protein. And the safety study in humans had 370 individuals, which demonstrated a pretty good safety. And then the important part is that they did a rhesus monkey challenge model. 72 monkeys here, and they challenged these, so they gave, what they did is vaccinated, and these are the different combinations that they used, and the one to focus on really is the blue line, because this was the most protective. So what they did is they vaccinated the animals and then challenged them with this SHIV. This is a chimeric virus, so it's SIV with an HIV-1 envelope, and the envelope is the SF162. That's an R5 tropic, mactropic envelope. So it's kind of a humanized SIV. So you can see, and what they did is challenge the animals, I think, weekly, and this is in this Kaplan-Meier analysis. You can see the percentage of uninfected animals, and so at the beginning, obviously, all of them are uninfected, but as they get challenged, pretty much in most arms, most of the animals become infected. But in this combination arm, you can see that they managed to protect about 2/3 of the animals out at 6 weeks. So that's pretty, that's quite encouraging that you can protect these animals from quite a pathogenic virus using this vaccine, this adjuvanted approach. This figure just shows you that there's a correlation between the immunogenicity and the protection. So this is on this scale you've got the human data, and on this side the rhesus data. So that's a very interesting and important, I think, observation that's going to be taken forward, I'm sure, in larger studies. Also, on the next, turning to transmission. I was quite lucky because I was running out of ideas for this talk sort of last week, and then we got this hit the news, so I thought this is great. We can put the partner study into this talk. And you probably all know about this. This was looking at MSM, who are on antiretroviral therapy, and looking at the risk of transmission. Of course, we've had the HPTN 052 showing that heterosexual transmission doesn't occur if you're on antiretroviral therapy, but there was a slight gap in the literature about what's happening in gay men. And as you've probably all heard, the number of linked transmissions was 0. So it's just quite a boring figure in many ways, but it's good, and it's a good boring figure, showing that there were no linked transmissions in patients who are suppressed on antiretroviral therapy and who were having condomless risky sex. There were transmissions in the study, but they were unlinked, and so suggesting that there were obviously there was infection from patients who were HIV positive who were uncontrolled into this population group, but they were not linked. So with 5 minutes to go, I think we can talk a bit about remission and cure. So this was the so-called Miami monkeys, I think, was of interest. This was published in Immunity by Ron DeRozier in the US. And essentially what this study shows is that, and remember, so the aim is to achieve remission, and what they did is they've taken this rhesus macaque model, and the virus they're using is SHIV. It's a slightly different one to the one I showed earlier. This is an AD8 expressing SHIV. That AD8 is also a macrotropic envelope. And they're trying to stimulate these monkeys to produce their own broadly neutralizing antibodies by delivering the genetic code for those antibodies using an AAV, which is an adeno-associated virus. So what you're essentially using is this virus as a vector to infect tissues in the monkeys and to then start producing significant quantities of monoclonal antibody. And so these were untreated monkeys that were that then received a vaccination, as you can see here, bNAb encoding DNA, intramuscular injection, and this figure is essentially showing you that antibodies were generated, but the problem in this study was that there were antibodies generated by the monkeys against those antibodies. So that limited the efficacy of this whole study. But nonetheless, you can see here, in this sort of cartoon, that certainly in a minority of the monkeys, they managed to achieve suppression of viremia, which is pretty remarkable, with 2 antibodies. So I didn't tell you the antibodies were, again, these 2 bNAbs called 3BNC117 and 101074. In fact, there were 3 antibodies. One was 10E8, but as you can see from this figure, the 10E8 antibodies really for some reason, there were very low titers, and it's probably explained by the fact that there were lots of host antibodies against this monoclonal. The other 2 antibodies were more successful in at least one animal, and that, of course, that's the animal that was able to control viremia here down to undetectable levels out to 240 weeks post-infection. So that was what they called a Miami monkey, which they think is functionally cured. These are the trajectories of viral loads of all the other animals in the study, and you can see that there's a sort of very modest reduction in viral loads. So I think this was an important proof of concept study. Again, only one animal was able to suppress virus. It seemed to be associated with good titers of 2 of the key bNAbs. So the questions are, why did this monkey managed to achieve these bNAb concentrations and the others didn't? Was it all about the host immune response, or was there something else? And can this be replicated, I guess? And just going to finish with obviously the London patient that was reported by our group in London. But I'm not going to talk about this because there's a talk in 2 days' time, and so I've only done 9. But I thought, in the interest of time, it would probably be advantageous for that. So I'd like to thank you all. But importantly, I've only talked about papers published. There were other really cool things happening this year. Of course, Atlas and Flare were discussed at CROI, and this is injectable ripivirine cabotegravir. But these are not published yet, so I didn't feel I could present them. But hopefully, in the talk next year, people will be talking about these really important studies. And the other thing I thought was really interesting is the Mellors group reporting at CROI some explanation for why you have certain patients with low-level viremia, even on fully adherent ART. They postulate that some individuals have. Sorry, is that? That's my wrap-up signal. So some patients have these clones in T cells which are able to generate virus that contributes to ongoing viremia. So that's their hypothesis. It hasn't been peer-reviewed, so again, a very interesting thing to look out for next year. So it's quite exciting already. Thank you.